What is the exposome? Different definitions of the exposome have been brought forward. Chris Roel, the former director of the International Agency for Research on Cancer, an agency of the World Health Organization, coined the term in 2005 and described it as a concept that encompasses all non-genetic factors and concomitant biological responses throughout the life course, affecting our health and well-being. In simple terms, where the genome describes all genetic factors, the exposome describes all non-genetic factors that drive health and well-being, such as lifestyle, diet, socioeconomic factors, and environmental factors, like air pollution and chemicals in our environment. In this MOOC, we'll use the term non-genetic and environmental drivers as synonyms. So why is the exposome important? We have several lines of evidence that tell us that the exposome is responsible for the majority of current chronic disease burden. We know this from twin studies, where researchers have calculated what the genetic component of the disease burden is. Think about migration studies, where the disease burden quickly assimilates to the disease burden seen in the country that people immigrate to. For example, in Japan, there is a low rate of colon cancer, but the rates become on par with the US rates upon immigration to the US. Or just think about the changes we have seen in the last decades, with a strong increase in people being overweight. All combined, these lines of evidence make us think that 70 to 80% of the chronic disease burden can be explained by the exposome. In 1986, the plan to map the entire human genome came to fruition, although at that time it was thought to be technically and economically unfeasible. The first draft of the Human Genome Project was successfully published in 2003, made possible due to several technical and computational breakthroughs. But if we know that the exposome is so important, why have we not done more to understand these non-genetic factors? So why the disbalance between genetics and exposomics? This is a valid question. There are many reasons, both scientific and political, but perhaps the main reason is that in contrast to genomics, we did not have the right studies and technologies to really measure all these factors. Face it, measuring the genome is not easy, but the number of molecules is limited to only 4 nucleotides: cytosine, guanine, adenine, or thymine. Albeit that we have many of those in sequence, means that each human genome is 6.4 billion letters long. Furthermore, our genetic code is stable and does not change over time, while our exposome is dynamic, with many factors changing over our lifetime. So, how do we measure the exposome? Well, there's no single method that would allow us to measure the whole exposome across the life course. Researchers are piecing it together by integrating data from many different sources. For example, it's now possible to measure and model many factors related to our home environment. Think about air pollution, noise, and green space, but also social factors like neighborhood cohesion, income, and our food environment. These factors external to our body are often referred to as the external exposome. In addition to the external exposome, we can measure the so-called internal exposome, which encompasses the measurement of exposures and their biological responses in biological fluids. With new biotechnological developments, we can now measure, for example, more than 3,000 chemicals in a single drop of blood. And not only can we measure these exposures, but also their biological responses, some of which can be found many years after the exposure has occurred. Piecing all of this information together is a daunting but doable task. Does that provide us then, similar as the genome, with a complete picture? No, it does not. It will provide us with a partial picture from which we have to use deductive techniques, including biostatistics and bioinformatics, to reconstruct the full picture. But what do we do when we have identified these factors? Well, to solve the problem, you first have to understand the problem. If we want to develop effective prevention programs, we need to understand the drivers of disease and their interconnections. Only with a proper systems-based understanding are we really able to combat complex multifactorial diseases like cancer, neurodegeneration, and cardiovascular diseases. Exposome research can provide such knowledge over time. Only then can we reduce the burden of disease and deliver on the promise of a healthier society for all. In this MOOC, we will further describe the exposome concept. How can the external exposome be measured and used? How can measures of the internal exposome be obtained and interpreted? How can we analyze and understand complex and large datasets? And how is this going to fundamentally change our view of public health and prevention? In this MOOC, researchers of the Utrecht Exposome Hub and the Dutch Exposome Research Program, Exposome-NL, will share their thoughts and insights.